Hey there, you're listening to the Syntalks podcast, produced by the International Genetically Engineered Machine team at the University of Toronto.
I'm Devaki, and I'm your host for this episode. I'm a third-year student doing a double major in cells and systems biology and global health, and I'm a part of iGEM, a synthetic biology design team here at U of T. Thank you for taking the time to listen to our pilot episode. Our goal is to educate you about synthetic biology and debunk the myths surrounding genetically modified organisms. We'll interview professors and graduate students about their cutting-edge research projects and the ethical issues they face, their opinions about genetically modified organisms, and their pathways to becoming the experts in their respective fields. We hope you find these useful and enlightening. So without further ado, we'll delve right into our first episode. Synthetic biology is essential for all types of research and all subspecialties of science, including medicine. For the purposes of this episode, we interviewed 3 experts whose research depends on synthetic biology: Dr. Kathleen Hefferon, Dr. Jennifer Mitchell, and Nolene Subramaniam. Dr. Kathleen Hefferon is a Fulbright scholar from Cornell University who completed her PhD in virology at the University of Toronto and is now conducting research in plant virus expression systems and their uses in producing plant biologics. Dr. Jennifer Mitchell is also a professor here at the University of Toronto, and her team explores how the genome is folded and organized in the nucleus and how this organization influences regulation of gene expression. And last but not least, Nolene Subramaniam is a PhD student at U of T, and she explores regenerative medicine and the dysregulation of blood vessels. There are varying definitions of synthetic biology. Here's what Dr. Hefferon has to say about the definition.
It's difficult to set a precise definition. I kind of think of it as a mix of engineering life. So genetic engineering is certainly a component of synthetic biology. I consider development of transgenic plants, for example, to be synthetic biology. Also, generating novel forms of biofuel, I think synthetic biology can be involved in that. Drug production, even development of a new biological pathway, let's say in bacteria, to produce energy by photosynthesis, or even to engineer plants to make them more nutritious. I think those are all different aspects of synthetic biology. So it's kind of an umbrella term, really. In
Engineering life. As powerful and intimidating as it may sound, this emerging field of science can have many beneficial clinical applications, from creating new treatments to building new tissues, designing vaccines, and engineering the genome. This is exactly what our 3 guests have in common. Their ultimate goal is to eventually take their research to a place in clinical medicine. We're glad to have you here. Here's our conversation with Dr. Hefferon.
I appreciate it.
All right, so in the most simplest terms, what exactly is your research about?
So my research interests lie in using plants to improve human health. I use plants to produce medicines such as vaccines, antibodies, and other therapeutic agents that can, for example, fight cancer. While some of the time I do make transgenic plants to do this, I'm also exploring a new technology known as genome editing, as well as using plant virus vectors, which we can produce pharmaceuticals cheaply and rapidly.
So what sparked your interest in this field?
I started out as a virologist, plain and simple. I was really interested in finding out how something as tiny and basic as a virus could cause so much destruction to life. So the more I learned, the more sophisticated viruses became to me. And once I started to get on top of how some of these pathogens replicated and expressed their genes, I began to realize that they could be harnessed in ways that are helpful and not harmful to the planet. So what keeps me going are the serious challenges we face today, such as food security, climate change, habitat loss for wildlife, pollution, emerging diseases. And my interests are increased by the feedback I get from people in developing countries such as Botswana, Bangladesh, Egypt. These are people who contact me and would like to use the technologies that we develop in the lab.
Why do you think there is so much negativity surrounding synthetic biology and GMOs?
Well, this is just such a great and loaded question. I think I could spend all day trying to answer this one. But I think we can parse it down into several major reasons that have caused some people to become so negative. First and foremost, I think people in general are not as tech savvy regarding science, food, agriculture, etc., as we would like them to be. And they're easy to sway through mass media or the internet or fake news outlets, so that people become unsure and uncertain about what the truth really is. I think secondly, anti-GMO activists have been very noisy and have influenced policymakers and politicians to be against this new technology. A classic example would be the people of Europe, where an anti-GMO movement was used to change regulations regarding GMOs, and as a result, African countries who desperately need new technologies to improve their agricultural productivity are influenced by their European trading partners who they would like to sell their products to. So if the Europeans won't buy African crops or African synthetic biology products, then Africa won't be able to produce them. And this is a real problem in a continent where climate change and increased in population growth is definitely are definitely major concerns. Importantly as well, scientists have not nearly been as good at communicating their research as they should be, and it's hard for us to get out of the lab and talk to people about our science. It's not something we're used to doing. We're not used to having to defend ourselves or to explain ourselves, and we really need to do a much better job at this kind of thing. So those are my main reasons, I think, why the world is the way it is against GMOs today.
This is a bit off of a tangent, but how you mentioned that one of the obstacles that we face as scientists is communicating to the public. So by what means do you think we can sort of overcome that obstacle? How can we start communicating these things to the public?
Well, I think some of the ways that I've certainly heard of and that are somewhat effective are having more outreach for scientists, or even having an outreach requirement in academia, so that scientists academics are aware that this is part of the job, that they must go out and spend X number of hours a month, or every 6 months, and communicate with people in some format. And that can be giving a friendly talk at a library, or I just gave a talk at something called Science on Tap, which was a pub in Ithaca, New York, and
nice.
People were drinking beer, and I chatted about my science, and it was a challenge for me to make it understandable and interesting, and it was a very relaxed and fun atmosphere because people were having fun, and it wasn't hostile by any stretch of the imagination. So it was a good experience for everyone. Other things we can do is we can write to politicians and policymakers and make sure that our voice is heard. Can write on blogs. We can tweet and Twitter and use social media a lot more.
Yes.
We can present ourselves in videos and different types of, every type of media that's out there for communication, I think, should be taken advantage of by scientists, and we just need to train ourselves to do so.
Mm-hmm. Yeah. So how do synthetic biology principles, techniques, and advancement help your research? Would it be possible to conduct your research without synthetic biology?
Oh, I definitely use synthetic biology to conduct my research. We changed the DNA of viruses and plants to make them produce medicines and more nutritious foods. So synthetic biology is the most important aspect of our research, and definitely I would not be the only person who would say that. I think you'd find many people, researchers at the U of T, who would agree with me that it is the paramount, most important type of research that we do. Yeah.
What are some of the ethical issues of your research?
So again, that's a really good question, and mostly we divide our risk analysis in terms of human health and in terms of the environment. So with respect to human health, we're concerned that our viruses that we engineer are not in any way going to harm anybody or any animals, or that they contain allergens or some kind of toxicity factor. So we have to test them and make sure of these things. So that's something that we always look at. Another thing is with respect to the environment, I think the biggest concern would be whether or not any of our engineered viruses could escape from the lab and somehow end up in the wild. And this is disconcerting, or on somebody's crop, for example. It is disconcerting, but there's many ways to mitigate this or to avoid this problem. First and foremost, viruses are made of components such as a coat protein and a replicase. So if you take away one of these, they're not able to replicate, and you disable a virus, it's not going to be able to infect a plant on its own. It's only going to be able to do so under laboratory conditions into cells that you have designed also to be susceptible to that virus. So they're just not able to get away very easily from the lab or recombine. They would not have the genes available in their genome to recombine and wreak havoc out in the wilds.
What are your thoughts on antibiotic-resistant bacteriophages?
So there are many, many types of bacteriophages. Bacteriophage is a virus that infects bacteria. So there are viruses that can kill bacteria, including antibiotic-resistant bacteria. So can we design bacteriophages so that they will kill off these bacteria that are antibiotic-resistant and of great risk for humans? And I think that the research is very exciting, and it's moving forward. It's tricky. It's not as straightforward as I'm presenting it. And I just know that from talking to people who are researching this, and it's a bit of a challenge to work with. But it is moving forward, and I think we will see, instead of necessarily new types of antibiotics, we'll see these other types of deterrents for these bacteria that are resistant to antibiotics. So it's a good one.
What is your message to students aspiring to become professors like you?
So I think that the most important thing for students to know is think of what they're learning as transferable skills that they can use in the future. So my feeling, for example, right now in the lab, I'm making a plant virus that is going to reduce tumor cells in cancer patients. I'm really excited about it. If you had told me that I was going to be doing that 10 years ago, or when I was working on my PhD in plant virology, I never would have believed you. It just sounds so unlikely and impossible. But here I am doing just that. And so disciplines that you are, the courses that you're taking today and what you're learning today, they're going to be mixed and matched in the most unusual and exciting ways in your futures. And you have to think more in terms of what you're learning cross-discipline and how you can apply it in different ways for the future, as opposed to necessarily sticking with the straight and narrow that you've learned up till now. So think of being able to jump easily from one discipline to the next, because you never know. We have now computer science and biology together we've made genomics and proteomics. It's very interesting for computer scientists to now know something about biology, or biologists to know about computer science. And we have artificial intelligence, which has all of a sudden come out. And some people are working in the biology interface with artificial intelligence, with really great and significant plans for the future. And I think there's all kinds of interesting things that will come out. So just think in terms of what skills are you learning, how can you transfer them easily in the future for whatever might come around the corner. That would be my advice.
So a majority of our audience is actually undergraduate students. And being an undergraduate student, as most undergraduate students actually look forward to landing research position in labs. So you're a research professor. What do you look forward to in a person when you're taking students in, say?
Oh my goodness, okay.
Or volunteer positions, work-study positions, anything.
Mm-hmm. Certainly if they have some basic lab skills, that would be great. And from my point of view, that means knowing, for example, how to run an agarose gel, knowing perhaps have had the experience of isolating DNA from bacteria or plasmid DNA or using restriction enzymes. So very basic molecular biology would be wonderful. It's not essential, it's not critical, but it's nice to have. It just means that if they're in the lab, I know that they will be able to work fairly independently or get up to snuff quite quickly, which is nice for everyone else around them too. So that would be the main thing, I think. Showing some interest and even coming with a project that they'd like to test out. I think that's so much fun. I'm never going to tell somebody no if they have something cool that they'd like to try out, as long as it seems safe and we have some parameters. I encourage that kind of thinking. So that would be really it. Those are the main things. Some basic research skills that they've learned in their courses, and persistence. If I don't respond right away, sometimes I get overloaded with work and I get behind. Just keep bugging me, and eventually we'll find a spot for you. So that would be, I think a lot of professors, you'll find that that's a problem, is we're just a bit too busy, and if you just keep bugging us, we'll eventually come around.
Once again, our goal is to educate you about synthetic biology and scientific research in general. As you may have noticed, our guests today and all the other episodes rely on synthetic biology techniques to conduct their respective research projects. We hope you understand that modern-day synthetic biology techniques is absolutely essential to conducting cutting-edge research nowadays. And this episode, and the next two, will show you why it is essential in the field of medicine. Whether it's designing vaccines using plant tissues, exploring the molecular mechanisms of dangerous diseases such as thalassemia, or analyzing exuberant blood vessels, and their applications in tumor biology. That concludes our first episode. To learn more about our team and projects, you can visit our website at igemtoronto.ca or check us out on Facebook, Instagram, and Twitter. Stay tuned to our next episode to learn more about exciting research projects in synthetic biology in the field of medicine. Till then, this is Adnan signing off from the Syntalks podcast. Goodbye.